So why people wear lab coats? To feel sciencey? To hide the fact you're wearing the same clothes as yesterday? Surprise the sponsor or safety inspector visit? Or if you're going to have a surprise sponsor or safety inspector visit, the surprise should probably be in quotes. It's too cold in the lab. Is it some labs everybody wears lab coats, some labs nobody wears lab coats, some labs some people wear lab coats. We're going to talk about helpers. It turns out that proteins, enzymes especially, are not that great at moving electrons around. And think back. Electrons moving around is a lot of chemistry. So they need help. Sometimes that help comes in the form of prosthetic groups, right, where minerals are actually integrated into the form of the protein itself. And sometimes the minerals just come in and help and go away. Is that they're a temporary buddy to help move sometimes particles themselves and a lot of times electrons. And those are cofactors. Is that a lot of dietary minerals are cofactors in various processes in our bodies. And then you have coenzymes. Now these are not enzymes. In fact, they come in a whole range of different molecule types. They're small. They're small enough that in order to absorb them, you don't need to break them down and take them in as pieces. Is that sometimes you need to play with them a little bit once they're in you, but they're generally absorbable in their coenzyme form. We make a bunch of them, and we get a bunch of them in our food. The stuff that we need, but we don't make, coenzymes, are what we call vitamins. And ideally, classically, by actual definition, vitamins are coenzymes that humans need to get from their diet. Is that they're not just the coenzymes. And when people talk about, plants need this vitamin or plants need that vitamin, is that it's kind of pointless. Is that one of the reasons why a lot of the vitamins we get are in plants is because they make those particular coenzymes for their own enzyme processes, and they just happen to have a bunch of them. We use them. Because we're omnivores, and we've been omnivores for a very long time, is that our list of coenzymes that are vitamins is a pretty long list, because we've got a bunch of stuff that we derive from animal-based food, a lot of the B vitamins and such, a lot of stuff we derive from the plant-based food, like the A's and the C's and such. If you're an animal that doesn't eat everything, doesn't mean you don't need those coenzymes. It means you better build them yourself. Is a cat has a process by which it builds the coenzyme we call vitamin C. Cats don't eat anything that has vitamin C in it, so they need to buy it to make it themselves. You can actually dig through our genetics and find the remnants of enzymes that we used to use to build coenzymes that weren't in our diets. Our diets have changed quite a bit over many, many million years. Our ancestors were much more plant eaters and much less animal eaters. So you can find degraded versions of the codes for enzymes to make, like, I don't know, I'm making this up, B12, I would think, for example. Why is it degraded? Because we haven't used it. So when it mutates, we don't have any reason to sort through the working mutations from the non-working mutations. And so eventually it winds up being a degraded code. It doesn't produce anything useful anymore. But what does that matter? We're not using it anymore. And sometimes other proteins will come in that are not really coenzymes, but they are necessary in order to help out. If they have a particular name, it's called a cooperative effect, and it's a complex effect but I don't really know whether those I don't know a particular name for them. Okay, one more item in this chapter, and then we're done with this chapter. This is the last stuff we're covering for the first exam. You have an enzyme doing something in your cells. You really need it to do it, and then you don't need it to do it for a while. So what do you do with that enzyme? Well, if you don't need to do it for a really long while, maybe you put the enzyme into a peroxisome, we'll talk about those later, break it down and reuse the bits and pieces, use the amino acids to build other things. But maybe you are going to use it fairly soon, and you just want to shut it off for a while. Shutting off an enzyme is called inhibition. And it turns out there's a few different ways to do this. Now, a lot of poisons work by shutting off critical enzymes and then screwing our chemistry up to the point where it produces a toxicity. Sometimes it'll kill us. Like a lot of fungal poisons screw up the chemicals that allow us to make proteins. Imagine that's going to affect cells all over your body. You have cells all over your body making proteins they need to make in order to work. And so a lot of things like mushroom poisoning, very generalized effects. Whereas other toxins may go after particular muscle proteins. Sometimes it's enzymes, and they're inhibiting the enzymes or turning them off. A lot of times so they can't be started back up again. And sometimes they affect other kinds of proteins as well. Those are not generally called inhibition then. There's 3 different approaches to inhibition. Sorry. One thing we're going to put up here. The inhibitors themselves can attach to the enzyme. Sometimes they're specially made inhibitors. Sometimes there are other things that attach up there. But they attach to the enzyme someplace, and that place is called a regulatory site. In the case of competitive or direct inhibition, the regulatory site is actually in the active site. There's 2 different types of non-competitive, also called indirect. Obviously, your substrate can't fit into an active site that already has something in it. Doesn't even have to fill it up. Just needs to be in there so that the substrate can't fit in the dent. Kind of like you go to park your car and somebody's left a garbage can in the garage. Well, you can't get your car all the way in there, and you can't park it because the garbage can's in the way. Doesn't have to be another car to be in the way. There's 2 different types here. Maybe the garbage can's not in the garage, but they went to put it alongside the door and it's sticking a little bit too far out into the opening. Not actually where you're trying to put the car, but you can't get past it to get into the garage. That's one type of non-competitive or indirect inhibition where the attaching the inhibitor, does something so that the substrates can't get into the active site. An allosteric effect is something that changes the shape of a protein molecule. We'd already talked about this with high temperatures and hydrogen ions and hydroxide ions, is that there's a bunch of different things. But there are inhibitors that will attach in a regulatory site, sometimes completely far away from the active site, but it changes the overall shape of the molecule, so the active site shape changes, and it doesn't fit the substrate anymore. And as long as it's on there, the inhibitor, the enzyme is shut down, even though the inhibitor may be nowhere near the active site. And there's also the term, reversible and irreversible. Do you want to shut the thing down permanently? If you're going to recycle it, you may want to just shut it off completely. A poison very often will irreversibly inhibit an enzyme. It doesn't care if it turns back on. It would prefer it didn't turn back on. But if you're going to use the thing later, you want to do irreversible inhibition. One more connected idea here is that you have a pathway. It's making a product 5, 6, 7 steps down. That product will fit in the regulatory site of the very first enzyme in the pathway. Not a great affinity, doesn't fit in there terribly well. It fits in, kind of loosely and shuts the enzymes down. But what happens is, the more end product you build up, the more it shuts down the first step of the pathway. So it's controlling its own production. You get a little bit too much of it, production slows down. Is it you start to use a bunch of it, production speeds up because it can no longer inhibit the first enzyme in the pathway. So that is end product inhibition. It's kind of an inbuilt control in the system. There must be some sort of factory analogy I could come up with for that, but I can't think of one. Okay, that is it for chapter 6. It actually says chapter 7 on my sheet. No wonder I said that wrong before. And as I said, this is the last of the first exam material.